be Paulo Sérgio Alves Bueno, and Paulo Sérgio will talk to us about the application of computational biochemistry techniques in the discovery of drug candidates in silico, in vitro, and in vivo studies. Paulo, are you there?
Hi, can you see me?
Yep.
Okay. Can you see my slides?
Yep.
Okay. Good afternoon. My name is Paulo. Thank you, young scientists, to program SBBQ and the SBBF for the opportunity. My presentation refers to my PhD work and it presents a collection of 4 articles produced in the period. The title is Applications of Computational Biochemistry Techniques in the Discovery of Drug Candidates in Silico, In Vitro, and In Vivo Studies. Computational simulations allow us to understand various aspects of the interactions between a cellular target and a ligand. In the example, we have exploited the docking interactions of some natural compounds as candidate for the treatment of hyperglycemia. In your lab, we use simulations for prospecting new ligands of biotechnological interest. Our targets are proteins, specifically enzymes. The computational simulations performed are homology modeling, molecular docking, and molecular dynamics. The ligands selected by these filters are purchased for experimentally test. The research protocol in your lab consists of the following steps: obtaining the 3-dimensional model of the enzyme with a ligand in the binding site, minimization of the protein ligand complex by the conjugate gradient method, build of ligand libraries for virtual screening, validation of molecular docking protocols by redocking, virtual screening of the libraries, repetitions in the docking program for the best results, repetitions of best results in other docking programs, in silico toxicity assays, molecular dynamic simulations, selection of the best candidates, purchase or synthesis of candidates, and finally, experimental validation in vitro or in vivo. We applied this protocol to 4 different problems using 4 enzyme systems: Chorismate synthase, Homoserine dehydrogenase, Alpha-amylase, and the viral cathepsin. The first 2 works have the specific objectives of to prospect drugs candidates for the treatment of paracoccidioid mycosis disease, or PCM. As a strategy, we prospect for inhibitors of the enzymes Chorismate synthase and the Homoserine dehydrogenase. So PCM is a systemic mycosis caused by fungi of the genus Paracoccidioides, where Paracoccidioides brasiliensis is the best known species. In Brazil, PCM is considered a public health problem and is a neglected disease. There are no specific drugs for PCM, and the therapeutic arsenal is limited to 2 classes of anti-fungal, polines and azoles. So our first work was titled New Inhibitors of Chorismate Synthase Present Anti-Fungal Activity Against Paracoccidioides brasiliensis. Our first target was the enzyme Chorismate synthase, which is the last enzyme of the Chorismate pathway. The Chorismate synthase converts EPSP and Chorismate to produce aromatic amino acids. Humans do not need this pathway, favoring selective toxicity. The enzyme is modeled by homology in the active conformation bonded to FEMN cofactor and the EPSP substrate. The first step before virtual screening was to validate docking protocol by redocking. The RMSD of 0.762 angstroms validate our protocol using Autodock program. The virtual library assembled contained 196,090 ligands. As a result of virtual screening, the CICS2 ligand was selected since it presented a better score than the substrate. The CICS2 has not yet been described as inhibitors of Chorismate synthase. The in silico toxicity study demonstrates that it is not toxic to human cells. In partnership with the medical mycology, we observed the concentration evaluated CICS2 was not toxic to mammalian cell lines up to concentration of 16 times the mic. The ligand also shows strong synergism with amphotericin B, the reference drug in the treatment of the PCM. Our collaborators from Graz University evaluated the binding of the CACS to Chorismate synthase, which gives us dissociation constant of approximately 20 micromolar. And the enzyme inhibition, say, gives a IC50, approximately 29 micromolar. These were the best binding parameters found for Chorismate synthesis till that time. The second work, we used the enzyme Homoserine dehydrogenase as target to prospecting potential inhibitors by similar methodologies. Homoserine dehydrogenase is an enzyme of the amino acid pathway and is absent in humans, which makes it an interesting drug target. The active form of the enzyme was ruled by homology in presence of the NADP cofactor and the homoserine substrate. 3 libraries totaling 112,572 ligands were assembled and screened by 3 different docking programs, Autodock, Molego, and CLC. The ligands HS7, 7, 6, and 9 were selection among the 3 programs. We carried out the simulation using the substrate homoserine as reference indicates that the enzyme ligand complex were stable. Then the ligands were purchased for microbiological assays, and the results indicate HS9 was the most effective in controlling the fungus growth. However, HS9 becomes toxic for mammalian cells at concentration 60 times the mic. Even so, it's a much larger window that described for amphotericin B. In the third work, we evaluated the binding of tannin derivatives to the pancreatic alpha-amylase in order to better understand activities hypoglycemics. So alpha-amylase are enzymes that break the alpha-1 for glycosidic bonds of start. Its inhibition prevents the increasing glucose and preventing hyperglycemia. This strategy already exploited by the anti-diabetic medicine known as acarbose, a commercial drug. Professor Rosanne and her student Camila described 2 tannin derivatives, inhibitors CTN and ATN. They have the property to inhibition alpha-amylase in vitro and decrease bond glucose levels in vivo, mirroring model. However, the details of interactions of tannins with the enzyme at the molecular level was missing. So we performed the homology modeling of the pancreatic alpha-amylase bonded to inhibitor A carbose pentasaccharide, validate the docking protocol by redocking, performed docking simulations using the program AutoDock and the Molegro. The common docking results presented the clusters of ligands conformations, suggesting a binding motif, and the poses of the best scores were in molecular dynamic simulations. Here we have the results of molecular dynamic simulations for E-carbose and the ATN. The rit maps indicate that the ligands moved a little along the simulation's time and stay bonded the binding site. Here for amylose and the CTN, the amylose ligand moved a little in the active site after 6 nanoseconds, but finds a pose and a place that becomes stable in the rest of the simulations. This also suggesting binding pattern. The delta G binding was calculated by IUPAB-SBBq. Delta G are in agreement with the inhibition constant obtained experimentally. This correlation allows us to infer that the ligand's possibility binding to the enzyme as indicated. So the last article in my presentation use work, the structure of viral cathepsin from Bombyx-Moore nuclear polyhydrosis virus as targeted against glycerine, docking, and the molecular dynamic simulations. The silk industry in the segment that produces silk and contributes to the economy of several countries. This activity turns around Bombyx-Moore, the silk producing insect or silkworm. The Bombyx-Moore nuclear polyhydrosis virus is the infection agent that causes the main loss in the silkworm creation. So this work is a special interest of the silk industry and is supported by the Bratak company. Here we have the silkworm life cycle. The caterpillar is infected by ingestion of virus. When the caterpillar dies, its integument is disintegrated by the activity of cathepsin and chitinase protease, thus releasing polyhydrovirus into the environment with source and the inoculant for infections of other caterpillars. So the objective of this work was to model the structure of viral cathepsin in complexity with a commercial cathepsin inhibitor, MIP, in order to use it as target for virtual screening simulations to identify inhibitors of this enzyme. Using molecular docking programs, 4 ligands were selected: BM1, BM2, 4, and 5. In the molecular dynamic simulations, it was possible to observe that all the enzyme ligand complexes are stable, and also to calculate the delta G, where the compounds BM2 and BM5, the better ranked. Experimental procedures were carried out with the collaboration of the group of Dr. Maria. It is observed that there was a decrease in mortality of approximately 50% with just a single dose of drugs. Other studies carried out later, where the dose and the concentration of BM5 were exploited, show a reduction mortality of up to 8%. So the final conclusion, Using computational simulations techniques, we were able to select candidates for specific enzyme inhibitors. The techniques used here point to more likely candidates. They demonstrate the sustainable of in silico methodologies as they reduce costs with chemical material, energy, and the time for experiments. Future studies with the recombinant enzymes Homoserine dehydrogenase and the viral cathepsin will be carried out to prove the enzyme-ligand interactions. I'd like to thank to the team of the Structural Biochemistry Laboratory, thanks to out-partnered laboratories, and thanks to funding agencies. Thank you all.
Thank you, Paulo Sérgio.